An enzyme, or group of enzymes, that's very important in our ability to manipulate DNA and recombine fragments of DNA pretty much any way we want to, are restriction endonucleases. These are enzymes evolved in bacteria as a way to protect cells from infection by bacteriophage. The DNA of the virus, once it got in the cell, if detected by a restriction endonuclease, would be cut up before it had a chance to do its damage. Restriction endonuclease is named because it would restrict phage growth, and then endonuclease means a nuclease that attacks a DNA polymer somewhere in the middle of the chain. Restriction endonucleases recognize very specific sequences of DNA, usually very short, 6 to 8 bases, or 4 to 8 bases actually, 4 bases long to 8 base long sequences, that they then bind to and cut. So here's our restriction endonuclease digestion of a piece of DNA at the 6 base recognition site. And as you can see, the digest was unequal. That is, it occurred at 2 different positions on either of the 2 strands of the double helix, generating what are called sticky ends. For the bacterium, that was enough to inactivate the bacteriophage DNA. We can use this phenomenon, the existence of restriction endonucleases, to recombine DNA at will. You can recombine DNA from any 2 sources, any 2 organisms. You simply cut them with the same restriction endonuclease, and then you mix the DNA from the 2 sources in this fashion, and you add DNA ligase, which will catalyze the stitching together of DNA fragments, and you end up with recombinant DNA. So in this example, we took a plasmid, it's a piece of circular DNA independent of a bacterial chromosome, but often housed in bacteria. These plasmids reproduce independently. But basically, they're little circles of DNA. If you cut them with a restriction enzyme, you get that one that produces sticky ends, and then cut some other DNA with sticky ends, or produce DNA with the same sticky ends. You can combine them. The complementary sticky ends will, we say, anneal. They'll come together. They'll form H bonds. And then if you use DNA ligase, you can make whole circles that now contain the various fragments you've added to the linearized plasmid DNA. So here's one blown up a little bit, showing the structure of this recombinant. So we have mostly plasmid, but it's been recombined with some foreign, in this case, double-stranded copy DNA, right, that was made with a reverse transcriptase, to which has been added these sticky ends, which we can do chemically.